Ja, okay, welcome. Very, very happy to see so many people, even in such a late afternoon session. But it's a very interesting topic we have today. And now we are moving into the Central Arctic Ocean, life at the extreme. And we have several interesting talks because we know so little about the Central Arctic Ocean. And now we're also going into a new decade where we have a big focus on the Central Arctic Ocean. And I was quite happy to hear the previous talks that now it actually will be an effort to see how we best can manage this area too, even if it's beyond the national borderlines in a way. It's an international sea. So we will start with an early career scientist. And you know, this is the area for the new generation to explore. We have barely started, or we have quite a few data, but we are really depending on the new generation to explore this deep ice-covered sea that is turning blue. So I'm happy to introduce Doreen Kolbach. She has been in Canada, she's been working in Norway, and now she's established in Germany, and she actually leading this Helmholtz Young Investigator Group, have now established with PhD and master students, and the title is Double Trouble, so that was a bit interesting. I hope that's not the case, but she will give us some insights into the underexplored Central Arctic Ocean food web, trophic interactions, and give us some insights from the recent expeditions. So please, Doreen, come on the stage, and we're looking forward to hear more about this that we know so little about, that you have been investigating. And you have not only done it in the Arctic, but also in the Antarctic. So actually you're quite well experienced already, polar scientists, and I'm happy you got this new established group. The floor is yours.
Thank you very much for the introduction. So my name is Doreen Kolbach, and I'm a researcher at the Alfred Wegener Institute in Germany. And today I would like to talk about or highlight our past research, and also talk a bit about some recent studies, which are all focused on the Central Arctic Ocean ecosystem and food webs within. And I would also like to take this opportunity to thank all the people that have been involved in the different projects, many of them from Norwegian universities and institutions, but also from Germany, the UK, and France, for example. So the Central Arctic Ocean has historically been understudied and underexplored, and that has many different reasons, including its remoteness and therefore limitation when it comes to fieldwork, particularly in the winter season, and therefore there are still many, many knowledge gaps surrounding the Central Arctic Ocean ecosystem structure and functioning. Over the past years, there have been more and more research projects, studies, initiatives, and so on that are trying to shed some light on the Central Arctic Ocean ecosystem, and they all have the same goal of creating a more holistic understanding of this understudied ecosystem for future sustainable development of this region, and also sustainable management approaches. And for that, we need to create more knowledge and a deeper understanding of the impact of climate-driven sea ice loss and warming, for example, but also the northward migration of invasive species and the impact of the rising fishing and shipping activities in the Central Arctic that will also come with a higher impact of anthropogenic pollution, which we heard quite a bit about already. Okay, and today I would like to talk a bit more in detail about a recent study that was part of the Norwegian Nansen Legacy project. And for this study, field observations and sampling took place in August and September 2021, and covered the Western Nansen and the Western Amundsen Basin, which form the Eurasian basin of the Central Arctic Ocean. In total, there were 5 main process stations, or P stations, P7 to P11 that you see there highlighted, and only the southernmost station P7 was sea ice free, and the other stations were sea ice covered during the sampling period. And in this study, we investigate and compare the status and underlying processes in the Nansen versus the Amundsen basins, and we provide an extensive inventory of physical, chemical, and biological components of the sea ice, the pelagic and benthic environments. And we did see basin-specific variabilities in all three environments, but they were most striking in the water column. So for this talk, I will focus on what we observed in the pelagic environment. So first we have a look at the hydrography. So we look at some seawater properties. In the Nansen and Amundsen basins, you have the 5 P stations on top, P7 to P11 from left to right. We saw a strong imprint of warm and saline Atlantic water masses at the near slope southernmost station P7, and that was also associated with stronger vertical turbulent fluxes, which ultimately supported and facilitated higher stocks of bacteria, protists, and zooplankton. I will get back to that later. In general, the Nansen basin was warmer and saltier in comparison to the Amundsen basin, and we also had, for example, higher chlorophyll A values in the surface waters. In the Amundsen basin, on the other hand, we saw signs of the transpolar drift that included lower salinity, and we also measured higher silicate and dissolved inorganic carbon concentrations in comparison to the Nansen basin, where we had higher nitrate concentrations. Then we move on to the pelagic protist communities. In the top panel, you see the abundances of the 4 main groups of protists, which were diatoms, dinoflagellates, other flagellates, and ciliates, and that's in the uppermost 90 meter of the water column. And then underneath, you see the vertical flux of these cells in the uppermost 500 meters. The protist communities and the flux were dominated by small cells, which were mostly flagellates, and that was the case at all stations. And we found the highest protist stocks, primary flux, and also net primary production at the southernmost station P7 again, and they were generally higher in the Nansen in comparison to the Amundsen basin. And then we also investigated meso and macro zooplankton communities. We found that in general the biotic stocks were higher in the Nansen basin, and then again particularly high at station P7 that you see on the far left in these plots, and that was the case for all meso and macro zooplankton groups. Fish was in general very sparse. There were only 7 individuals called in total 9 hauls. And we did see some signs of potential species range extension. For example, the helmet jellyfish and Greenland halibut larvae have not been reported that far north before. But of course, that needs some more in-depth study and confirmation. And then we also used carbon and nitrogen stable isotope values to recreate the food web structure in the 2 basins. The isotopic compositions indicated a typical food web structure found in Arctic ecosystems. So we had decreasing carbon stable isotope values from sediment, particulate organic matter, which is in this brownish beige color. I hope you can see that, I'm not sure, via sea ice associated particulate organic matter, which is in blue, to pelagic particulate organic matter in green. And then we also isotope values in the benthic invertebrates in comparison to the pelagic zooplankton, which reflects the highly reworked organic material we find at the seafloor. But there was also a strong overlap in nitrogen stable isotope values between pelagic and benthic invertebrates. And then when we have a quick look at the pelagic zooplankton at the different stations, we found that there was a strong overlap in carbon-stable isotope values between different zooplankton groups, and there was also the case across multiple trophic levels. And this pattern was also robust across all 4 sea ice covered stations, P8 to P11 that you see here on top, indicating similarities in food source use for those species. And then again, station P7, the sea ice-free station, was very unique. The pelagic zooplankton caught at this station had lower nitrogen and carbon-stable isotope values in comparison to the sea ice-covered stations. To sum up the study, so we found that the pelagic primary production, as well as microbial and faunal stocks, decreased from the sea ice-free station just north of Svalbard into the Nansen and ultimately into the Amundsen Basin. And that was likely the result of the lower nutrient availability, stronger stratification, and reduced light, as well as potentially seasonally declining light levels we found in the Amundsen Basin. The distinctly higher biological activity at station P7 indicated that for the future, potentially deployed monitoring stations just north of Svalbard would not be representative for the Nansen Basin itself, let alone the entire Eurasian Basin. So therefore, there's a strong need for a regional approach in regards to future Central Arctic Ocean management strategies. So we did learn quite a bit from this study, but of course there are still many more knowledge gaps that need to be filled within the next years and decades. And we're trying to do that, at the moment at least a little bit with my Helmholtz investigator at AVI, which is called Double Trouble. Impact of global warming and anthropogenic pollution with a focus on the Central Arctic Ocean ecosystem. We are using different state-of-the-art, mostly trophic marker methods to study food web interactions and dependencies in the flow of carbon through the food web. And we have a strong interest and focus on understudied species, which includes squids, mesopelagic and gelatinous zooplankton. In the current master's project, we are utilizing long-term seasonal abundance data. That's 16 years of gelatinous zooplankton in different Greenlandic fjords. That's part of the Greenland Ecosystem Monitoring Program. And we hope to also extend this research to the Central Arctic Ocean in the future, where the role of gelatinous zooplankton within the food web is still very much unclear and unknown. For this study, we're also using stable isotopes from different fjords. This is an example of Chetoknats showing dissimilarities in food web structure and food source use of Chetoknats in different fjord systems. And in the next step, we will also do that for gelatinous zooplankton from the Central Arctic Ocean, and that will actually be a starting point for a more detailed analysis on how they are feeding and what they are feeding on with DNA metabarcoding in the next step then. We also have a strong focus on the importance of sea ice algae-produced carbon as a food source in comparison to phytoplankton-produced carbon, and for that we are using different trophic marker methods. For example, we are looking at fatty acids and stable isotopes, but also so-called highly branched isoprenoids, or short HBIs, and those are lipid metabolites that are assumed to be exclusively produced by sympatric and pelagic diatoms, and they can tell us a lot about if a species feeds on sea ice algae-produced carbon or is utilizing sea ice algae-produced carbon. And in this example here on the bottom right, we're looking at carbon and food sources of pelagic krill species and found, for example, that Meganyctiphanes norvegica is utilizing sea ice algae-produced carbon to a great extent. In another current study, we are investigating the sub-ice algae Melosyra arctica that can grow in very long strands that are loosely attached to the bottom of the sea ice. It can grow very patchy and dense, and when the sea ice melts, those strands are released into the water column, and since they're very heavy, they sink rapidly to the seafloor and have been observed to serve as a food source for invertebrates we find at the seafloor. But so far, there's almost nothing known about the biochemical makeup of Melosyra and its role as a food source for sea ice-associated craters. So that's what we focused on in this study. We were looking at fatty acid stable isotopes, also highly branched isoprenoids, which were not produced by Melosyra, and also at sterols. And we found that the trophic marker composition of Melosyra was very typical of that of a centric diatom, but we did see high levels of the sterol camper sterol in the field samples, which we did not observe in the pelagic and sea ice-associated particulate organic matter from the Central Arctic Ocean. So that could potentially be very unique to Melosyra and could be used in future food web studies to trace Melosyra through the food web. And we also did some experiments with Melosyra using the sea ice amphipods Afarusa glacialis and Onesimus glacialis, and found that they were heavily interacting with Melosyra assemblages, and there were also various degrees of Melosyra consumption. In the second part of my Helmholtz project, we are focusing on anthropogenic pollution, and at the moment we are running a master's project which is focused on microplastics. We are looking at the presence of microplastics in stomachs and also muscle of mesopelagic fish from the Central Arctic Ocean, which is Benthusema glaciale, and then we compare that with the mesopelagic fish from the Southern Ocean, Electrona antarctica, and those are some very first and new results I showed today. So we did find microplastic particles in the fish in different quality and quantity, as you can see, so very different between the two fish species. And we also observed a high variability in polymers between different individuals of the same fish. We are also running at the moment a multi-stressor experiment. We heard a little bit about that from Kwang already. It goes in the same direction. So we are interested in the impact of warming and the presence of microplastics and PFAS, which are per and polyfluoroalkyl substances and are part of the forever chemicals on one key zooplankton species in many Arctic food webs, Calanus hyperboreus. And we want to know the impact on Calanus hyperboreus survival, hatching success. We also look at lipid sac size and on different enzymatic endpoints. And we have some first results as well I would like to show you today. The first is on the survival probability over time. So that's over the past 6 weeks we have been running this experiment in the different treatments. We found the highest and most stable survival rate at 1 degree, which is in this light blue that you see on top. And there seems to be a somewhat stronger impact of microplastics and PFAS in the cold temperature at 1 degree in comparison to the warmer treatment, which is at 4 degrees, if you look at the difference between the blue lines and the red lines. And then also the last plot I would like to show today is on the cumulative egg production over time in the different treatments. So that's after week 2, 4, and 6. We saw a low and steady egg production in the cold treatment, again, the control group, which is in light blue on the far left. In the warmer treatment, there was a somewhat higher egg production. That's the light red color. And then when we look at the single stressor, so that's either under PFAS or microplastic presence, there seems to be a higher hatching success, which could be a stress response. So it has been reported for this species before that they can release their eggs quicker when they feel stressed. But then when we look at the multiple stressors, including warming and microplastics and PFAS, there seems to be a lower hatching success. So maybe they feel too stressed, and we don't know yet. We have to still figure that out in more detail over the next weeks and months. And with that, I would like to come to an end and just point out some potential future research directions. So we need to know more, of course, about the baseline in the Central Arctic Ocean ecosystem. So we need to know more about zooplankton and fish stocks we have there, and how these species are interacting with each other, to do some ecosystem mapping, and have a strong focus on rare species and also invasive species, which will be very crucial information for future sustainable management of this ecosystem, and also to inform the Central Arctic Ocean Fisheries Agreement. And then there should also be a stronger focus on multi-stressor assessments, to create a better understanding of cumulative impacts, including sea ice loss, warming, invasive species, increasing noise level, increasing anthropogenic pollution, ocean acidification, and all of that. Yeah, and with that, I would like to end, and thank you very much for your attention.
Hi, nice talk.
Thank you.
Do you think the Periphylla, is that a new species because you are observing it now, or do you think that it's actually been a migration? And what is the environmental driver for this migration?
Yeah, so I'm not really an, I would say, expert when it comes to that. Maybe Sanna, I don't know if she's still in the room, she can say more about that. I want to, like I would say, just because maybe it hasn't been observed or reported before, doesn't automatically mean that they haven't been there before. So it could be like different sampling strategies, for example, that they haven't been caught. Yeah, that could be a reason. But then of course, warmer temperatures, and they go for it. That could also be a reason.
We have 1 minute more, so a short question, if anyone have. You want?
Then I will take the opportunity. Very, very interesting talk, Doreen. And this focus you have on these multi-stressor investigations, I think it's a core thing. And I was wondering, because now you had microplastic and or we have seen these studies from the ice that is quite abundant there.
Yeah, it can. Yeah, exactly. It can be. Then, of course, if the sea ice melts, it can be released into the water column. We actually don't really know about the Central Arctic Ocean yet. That's the main problem. So we took values from like Svalbard waters and so on. What has been reported there before. So that's what we base this experiment on.
Okay. Thank you. Yes, I forgot to introduce myself. I was so too eager to get into the talks. So my name is Janne Søreide from the University Center in Svalbard, and the leading chair here. This is Liz Jørgensen, and she also very well into the topic since you have been leading this Vega ICA report that just come out now in November. Institute of Marine Research. We will move over to a very, very exciting topic, very hard to sample, so we know very little about it. But Mats Granskog, a senior scientist from the Norwegian Polar Institute, he has actually dared to do this, and he was PI for the project Havok that took part in the big Mosaic expedition that you probably have heard about. And he will now tell us more about these sea ice ridges, that they are important but neglected, and maybe you will show some examples why they are neglected. And looking forward to hear more. Mats, please, the floor is yours.
Okay, thank you, Janne. I must admit that I feel a bit misplaced in this room because my background is physical science and I'm trying to talk biology to an audience that's way more clever than I am on this subject. So I apologize if this is being way too simple for you, but it's enough complicated for me. So I will deflect any biological questions to a few of you in the audience who actually were in the project. I'm looking at you, Philip. But yeah, so I can't take, this was really a team effort on Mosaic, and I will have this group. So of course, most of us have ventured out to the sea ice in the Arctic, and when you go there, it's not a flat sheet of white as you might see from far away from space. When you actually walk on the ice, it's very uneven and you have these ridges. And as an observer on the surface, you see a very small part of the sea ice. Anything that's below, of course, we miss. We don't see what's below, and 90% of the sea ice is actually below the surface. And we know from studies that up to half the sea ice volume in the Arctic could actually be ridges. So it's actually a big fraction of the Arctic ice pack. On a planetary scale, it's actually important. It's been shown in many modeling studies that the roughness they create, either on the atmosphere interface or the ocean interface, actually affects large-scale circulation to a big degree. So they're actually also important larger scale than themselves. There's been a handful of studies that's kind of shown snapshots that they could also be biologically very important, which I'll show you a few examples on later. From a human perspective, they are actually the most difficult part of the ice pack when you navigate with ships or if you build any structures in ice-covered waters. It's the ridges you need to take care of, even if they would show up once in a decade. You need to dimension your things to the forces these exert on structures. And as Janne kind of hinted to, people are lazy, so people have been studying the thin ice in the Arctic, and basically we have no data from ridges except for a few studies. And for Mosaic, we had the chance to go out for the whole year and try to do this, or that was our ambitious plan at the beginning, that we would manage to do this, but okay, life is not fair. So we managed to do something, but not as much as we wanted. So a quick 101 on ridges. This is a very idealistic picture of kind of an inverted pyramid and a smaller pyramid that is on the surface, and this is what you see, but as said, 90% of the ridge is below the surface, and it's completely invisible to most people unless you go down and dive, as Håkon usually does for us. Perhaps the interesting fact is that when they form, it's actually a maze of ice blocks with water in between. So it's like you would pour Lego bricks into a can of water, and it's kind of a very difficult maze to navigate and obviously very difficult to sample. But they are not static either. I mean, with aging, they also change, that there's more ice growing in them. They are covered with more snow, and some of these voids freeze eventually. So they're not that static either over time. And I wanted to highlight a few things we discovered during Mosaic when we had the chance to sample these from winter, spring, and in summer. And one is perhaps what I would call a winter surprise. And then we can show that they're really an excess habitat that provides a lot of excess space for the sea ice habitat. And we also find that they were a summer hotspot. And now I come to the difficult part where I don't know the background in very much detail, so please bear with me. But there's evidence that there's still something going on in the polar night, but there's no primary productivity. And there needs to be an energy source. And Lasse Olsen in our team came up with the idea that, well, what happens when you create ridges and all these ice blocks are submerged? So what happens is that this level ice, the ridges are formed from, is usually very cold in a winter, so it's actually trapping material that's inside it. But at the time you form a ridge, these are submerged and they kind of reach their melting point almost, or the seawater freezing point temperature, and then these ice blocks become very porous and any material inside them can be released to the water column, which again can be used for pelagic production. So that was something we didn't expect, but I mean, when you think about it, that's prone to happen. And level sea ice after the summer can contain a lot of organic material, and this way it could potentially be actually released to the water column. Then there's many microhabitats in ridges which you don't find in level ice, and these produce actually way more area for ice-associated organism. So we did kind of back of the envelope calculations that there's this ice block surface area is easily 20 to 30 times larger per unit area. And also the brine channel system in these ridges are much larger than in level ice. So even with a 5% aerial coverage of ridges, they provide the equal amount of ice surface. But in reality, usually have 30% coverage of ridges, so they might be hugely important. And then you have the water-filled voids. And then in some of data from both level ice and ridges on biomass, standing stock, and based on what we found on Mosaic and using what we found the ridge coverage was around the Mosaic ice camp in July, up to 80% of the ice algal biomass would reside within ridges, which is kind of a big fraction and something we don't usually sample because it's so bloody difficult. One. But I'm about to conclude, so I hope that you got excited that much that next time you go out on an ice floe in the Arctic, you forget the boring level ice and drill in a ridge. Thank you.
So 1 minute left for a very, very short question while the next speaker make herself ready. Any questions? Then I think we move on. I'm sorry. Next speaker, thank you.
You are a good timekeeper, Liz. So, yes, so our next speaker is Vanessa Petuzzi, and she's from UiT, the Arctic University of Norway, and she will tell us more about who is living inside sea ice, and done a quite extensive studies in Svalbard, and now also get some samples from the Arctic Ocean, and it's a very, very exciting story, so I'm looking forward to hear Vanessa. Please, the floor is yours.
Thank you, Anna. I'm going to be talking about the sympagic nematode Thuristos melnokovi. So we'll go a bit larger than bacteria, but we will stay small. For anyone that doesn't know what a sympagic nematode is, sympagic essentially means ice-associated, so we're talking about microscopic worms living inside of sea ice. And we know 5 species from Arctic sea ice, but we will be focusing on Theristos melnikovi specifically. It was described in the late 1980s, but despite that, we still have very limited knowledge about its actual distribution within the Arctic, feeding ecology, its dependency on the ice. We find it both in ice over the Arctic Ocean as well as fjord ice, and we don't really understand the full life cycle yet either. The main results in this presentation are from 3 fjords on Svalbard. We have Van Mayenfjorden, Wiedefjorden, and Wallenbergfjorden. But as Janne mentioned, there is also a note on results from the Amundsen Basin from a cruise in 2024. We still know very little in terms of species about actually which fauna lives inside the ice. So even though maybe nematodes are not what you fancy, there's many other meiofauna that can be worked with. And as we all know, those areas are fast changing, sea ice is declining, so it's very good to know who might be at risk or whether they're a winner or a loser. Very quick on methodology if you've never sampled sea ice. It's either ship-based if you're working over the Arctic Ocean or land-based with snowmobiles if you're working on fjordic ice. We collect sea ice samples with this COVAX ice corer. This is what the sea ice core looks like. We collect the lower 10 centimeters usually. That's where most of the biological activity is located, especially if we're talking about fjordic ice. Put it in a plastic bag, melt it, and then split the sample for molecular analysis, so an ethanol, and taxonomic analysis in formaldehyde. This time we also sampled in the late summer from Helmer Hansen, went on a small boat, collected different species of macroalgae, processed them, and again, ethanol for molecular analysis and formaldehyde for taxonomic analysis. So took all these samples, looked through them, picked out every nematode that I could find that was a Theristos, placed them on a slide, under microscope took pictures. For the taxonomy, they stayed on the slides. For the molecular analysis, they went into individual tubes, and we sequenced the whole 18S, and then built phylogenetic trees and haplotype networks based on that. Maybe none of you here work with nematodes, but please don't take any pictures. This is the last paper for my PhD that I'm hoping to publish. So if you've never seen a haplotype network before, it essentially shows genetic variation across geographical regions. We have 2 outgroups, Theristos agilis and Theristos levensis, which are benthic nematodes living mainly on the in sediment on the European mainland. And we have Theristos acer, which is the most closely related benthic nematode to Theristos melnikovi. And here you find the genetic mutations, so sort of like the genetic distance between. So we have one main haplotype identified through our analysis. It's the largest, with the most individuals represented from different regions of the Arctic, mainly the White Sea, the Kara Sea, and Svalbard. From that, 2 mutation steps away is haplotype 2 of a Theristos melnikovi from the White Sea. Again, nothing too unusual. But then what was quite interesting is that the 3rd haplotype was a Theristos melnikovi from the White Sea and what we had assumed to be Theristos acer from the macroalgae. But if it was a true Theristos acer, it wouldn't sit in the cluster of the ice nematodes, but somewhere down here or closer, which got us thinking, is this actually are these individuals of ice nematodes found on the macroalgae? For that, we cannot just trust DNA, and I hope none of you do. No offense. Old-fashioned taxonomy. So picking individuals of males and females and juveniles, taking the pictures and taking a lot of morphometric measurements to determine whether they are the same species, different species, or just different morphotypes. This is what I'm still working on, so if you want to find out, you will have to read my paper. And about the Arctic Ocean, from the Amundsen Basin, 12 ice nematodes in total were found, of which 8 were identified to be Theristos melnikovi. mostly within the range of adult sizes. And the sequence data is in work, and I think it will be really interesting to see where they fit within the haplotype network, to see if they're their own haplotype or whether they're mixed within the 3 established ones. But to obviously bring this into context, what does that actually mean? On Svalbard, what we hypothesize is when sea ice melts, obviously a no-brainer, everything melts out. But the question always was, where does it go? So Thristos malnukovi can just live out the ice-free periods on different species of macroalgae, lots of food there, other bacteria, other kinds of detritus, great habitat. And then once the sea ice starts to form, it's scavenged back up into the ice where males and females meet, they make babies, they reproduce, and then the whole cycle repeats itself. What does that mean for the Arctic Ocean though? So here we can look a little bit at Wallenberg Fjorden, especially the outer part is a sort of model fjord. It's connected to the Hinlopen Strait, and with that, to some degree, connected to sea ice export from slightly more northern latitudes. So we have previously sampled shoals of ice that have drifted in and found Thristos malnukovi there. So the question is, could the macroalgal refuge apply to Arctic Ocean Thristos malnukovi? So even if the sea ice melts, there is sort of refuge to be found and an alternative habitat to be found for them where they can survive. So that is the question we're currently working on. Just an acknowledgement to everyone that supported this research, and if you have any questions, don't hesitate to ask. Thank you.
Thank you very much. Great talk. And we actually have 3 minutes. So, questions.
Thanks for a very interesting presentation. This Melnikovi was obviously named after Igor Melnikov many years ago. He was a Russian fellow diver who I know well. But there must be some information of its distribution, since this has been known for quite a while, the species, and especially in the Russian literature, of what is its distribution in the Arctic Ocean and its marginal seas.
There is to some degree. I mean, we know we find it in the Arctic Ocean, Svalbard, definitely the Russian Arctic. When they do translate the literature, I mean, you can Google translate it yourself when you find it. And also the Greenland Sea, but it's quite limited because people don't usually work with meiofauna. They don't work with nematodes specifically. So its occurrences are limited. What I will say though, in recent years, the Russians have been putting out a lot of literature, but it's limited to their part, or that part of the Arctic they work in. So it is known, but it's still very patchy considering it was described in 1986. No worries. Thank you.
Thank you so much, Vanessa. And just to say again that Vanessa is one of you that actually have been studying sympatric meiofauna in Svalbard landfast ice. And I think it's super interesting, this comparison with the Arctic drift ice, because are we losing them or do they live somewhere else? So very tedious work to answer these questions. So that's why it's not so very well known. We will move over to my colleague, Anna Wader at the University Center in Svalbard. Oh, you're not? Okay, sorry, I'm jumping. So you have to wait a little bit, but now we have another very exciting talk. And there was someone talking about periphylla previous year, and I think Sanna will be the person to answer this question, because Sanna has done extensive studies on gelatinous zooplankton in a changing Arctic Ocean. And she is saying to me that there is many more species, it's not just these 2. And I said, oh, okay. So now I'm very interested to hear, Sanna, you are from Aquaplan Niva and you have a whole group here that's helping you with this, and I'm looking forward.
Thank you, Janne. Hello, everyone. Together with my colleagues, I would like to take you a little bit on this, what Doreen also mentioned, that is highly unknown and gelatinous part of the Arctic Ocean. But I take you a little bit back in time. 15, 20 years ago, there was a heavy discussion about increase of jellyfish globally, that our oceans are turning into slime. And we saw a lot of these kind of images where there was a heavy increase in jellyfish biomass, as an example here. A lot of this idea was coming from papers, like Claudia Mills' paper in 2001, where they were discussing that due to the human activities, there might be changes in the oceans, and these changes in the oceans are potentially causing areas that potentially have new blooms, of jellyfish blooms occurring. She also continued that the issue is not so simple, and in more cases, we actually don't have enough data to show this. We started to see more of these kind of data sets where they actually show the fluctuations rather than a constant increase in the few time series that actually existed. The main problem is that normally gelatinous organisms are not well sampled in traditional zooplankton sampling. So we do have very little data. Still stating this, that after this, in 2012 about, this started to be questioned, mainly because we do not have data. But recently, these kind of headlines start to come more in the Arctic. Jellyfish might dominate the future Arctic Ocean, and there might be a huge invasion of jellyfish into the Arctic Ocean. But what do we really know? Do we have enough data now to actually talk about these kind of things? I'll show you a few examples. Today, our knowledge of gelatinous organisms is mainly focused on few species, of which Berifula often being the common one. Our image of the jellyfish diversity in the Arctic is looking something like this. Few species that are kind of robust enough to be in our sampling, often be large ones that are easy to work with. So in the last years, we've been having extensive amount of cruises, starting from Nansen Legacy, also in the Arctic Ocean, to actually look how does the jellyfish diversity look like in these areas. And there has been many people picking up these samples, and I want to thank everyone who has been doing this hideous work and putting these bits and pieces of jelly together. So what we can see, that the more we sample, the more diversity we still detect. And we are still far from the numbers what we can pull out from the literature that has been ever reported in the Arctic. We've been also having several projects as part of the Norwegian Taxonomy Initiative to actually look at the jellyfish diversity, map which species we have and where in Norway and the European side of the Arctic. And big part of this is also not just to morphologically identify them, but also to develop DNA reference libraries. Because if we look at here, what are the public reference libraries for gelatinous organisms in different genetic markers, and the dark colors are the ones for the Arctic, we are missing a lot. So in these projects, we have had gentle sampling processing. This is very important. The key is also to work them on live and to be able to morphologically identify them, not to preserve them. And we've been also barcoding different markers, so different gene regions, starting with the Sanger sequencing and now going more into genome skimming to actually have a larger area of the DNA. So in total, we have worked up more than 5,000 specimens of jelly lops, and this includes more than 500 jellyfish species in this data set, and it's constantly growing. So basically what I was showing that the Arctic looks like this, it starts to look more like this. And we see a lot of diversity. We see a lot of smaller species that are not so easy to work with. But if we think about the future directions, where can we use this kind of a data set? There are multiple ways of using it, and I can show you just a few examples what we've been working on. One thing is to look at the species identification and systematics. Here we can see a species that is often reported in the Arctic, and it goes with the name unidentified Syripid tenophore species 2. But is this species 2 the same as, for example, Janne species 2? It makes it really hard to compare. So together with integrated taxonomy and actually working globally in collaboration, we are currently describing 5 new species to science, new genus, and potentially also a new family, which then allows us to actually look more which species are where, how are they distributed. When we have more and more data, we can then look what are the areas that they prefer, what kind of environmental conditions, and how do these change in the future, rather than working with the species number 2, where it's actually a big group. Another one is that we have this constantly ever-growing use of DNA metabarcoding and use of eDNA, environmental DNA. But we need something to actually match our sequences with. One is that if we look here, we can, in the table or the figure, we can clearly see that if we use traditional tools and in like a POLD, which is a typical reference library, we can see that we end up with the Syripid tenophore 1, Syripid tenophore 2, and these kind of names without being able to give them name, like proper species names or understand where they are. And this kind of creates a high uncertainty. Where are the species occurring? Are there any changes? But with this kind of combining morphology and compiling the DNA, we can actually create a DNA library which is curated, and we can come up with the proper names, which allows us to actually detect changes and who is where. Another one is that jellyfish is often, some of the taxa is having a very unique life cycles, connecting benthic and pelagic systems. So who of you could actually connect which of the pelagic stage is connected to which one of the benthic stages? This has been a problem for ages, and often they are identified separately, so that they are already having very different names. But with these kind of data sets, we can actually combine these things and start to understand the connectivity between the benthic and the pelagic systems. So with this, I think there are many ways that we can start looking and using and understanding the jellyfish diversity in the Arctic. There is a lot to learn, and there's a lot of areas. This is just about diversity, but in most cases, we don't know about their role, we don't know about their distributions, habitat favorites, or any of this. So it's hard to say whether the jellyfish are increasing in the Arctic. I think we don't have enough information. At the same time, it's important to think about what do we mean by the term jellyfish. Is it just the biomass of jelly lump, or is it actually diversity, where some species might be increasing, whereas others are getting extinct? So with this, I hope that we can collaborate this further and get more information in gelatinous zooplankton in the Arctic, and thank you.
Thank you so much, Sanna. And this link back to what are the sensitivity of these species when we don't even know that they are the same species, those living at the bottom and those in the pelagic. So are there any questions? We have time for 2 quick ones, or 1.
Very good talk. So you mentioned that I assume correctly that we need to link the morphology and the taxonomy with the genomics. But one of the difficulties, as you also mentioned, is to sample them. So how do you have any good tricks to sample them so that you can actually do the taxonomy? And
I think that question would be actually go to the people who are on these cruises to pick up them. I would prefer, we have in Nansen Legacy, we developed a protocol. Of course, it's not optimal because there's not limited time in the cruises. But what we often do is that we pick them up and take images. And these images already helps out a lot. Another is that you just pour your zooplankton sample and take an image of that. It gives us more information than not having anything when we preserve them. and combining different tools. So not just plankton nets, but also trawling and other tools as well.
Okay, I think we have to move on. I'm looking at the red screen here. That's over time. So then I will introduce my colleague Anna Wader, and we will continue with the molecular approach, because many of these species are also very, very tiny, so we can't actually see them that well. So Anna, she will look at or learn us about this microbial biodiversity of the Nansen-Amundsen basins. And we are looking forward to learn more, Anna.
Yeah, I'm Anna. I'm from University Center in Svalbard, same place as Janne. And I'm going to talk today about microbial biodiversity of the Nansen and Amundsen basin, and on behalf of my colleagues that are listed here. Let's see. So, why do we care about marine microbes? First of all, they make up a staggering 70% of the biomass in the ocean. So even though they're tiny, there's a lot of them. Also, they perform, I think somebody else said this already, but 50% of the Earth's primary production. So they convert inorganic carbon to the organic carbon that the rest of us depend upon. They make up or have representatives of an overwhelming part of the phylogenetic diversity that we know about on Earth. And that means that they can do a lot of different things, and they are important for a lot of the biochemical cycles of the ocean. And what I'll talk about today is protists, or unicellular eukaryotes. And in the Arctic, they are pretty much, or at least in the Central Arctic Ocean, they're pretty much the only primary producers, since cyanobacteria are pretty much absent. But even though they are important, there are still a lot of things that we don't know about them. We don't know necessarily who they are. This is especially true for the small flagellates that we can't really distinguish in a microscope. We don't know how they respond to their environment. We don't know how their communities are structured. And we don't know who are the active members of the community. I mean, I'm not saying we don't know anything, but there's a lot that we don't know. And as we've heard from several speakers, this is important to know both as input to management, but also to be able to see what is happening in an environment that is changing. So I think Doreen introduced this cruise nicely. So we did a transect up here in the Central Arctic Eurasian part of the Central Arctic Ocean, and this consists of the deep Nansen and Amundsen basins that are separated by this subsea mountain range, the Gakkel Ridge. These are similar environments in some senses, but they also differ in that the Nansen basin here is more influenced by, or directly influenced by, Atlantic water inflow, while the Amundsen Ridge over here sees more of this transpolar drift that brings sea ice and surface water from this part of the Arctic over here, from the shelves. What we sampled was this transect in late summer 2021. And in the red stations here that Doreen introduced, we sampled both sea ice and the whole water column down to the bottom. In these yellow stations, we only sampled the water column and then only down to 1,500 meters. And what I will present today is the metabarcoding data, so genetic data from a marker gene. And this gives us the genetic equivalent of a species table. And we sequence both RNA, which represents the active community of protists, and we also sequence DNA, which is, in addition, the active community. It's also dormant organisms and even dead organisms. So to the results, this is an NMDS plot, and each dot here is a community, and if the dots are close together, it means that the communities are similar. So we see, first of all, that the sea ice here in the boxes communities from sea ice are very different from the pelagic communities, and also we see that the pelagic communities really differ. They're significantly different based on which depth layer they were sampled from. So the euphotic communities are different from the mesopelagic, which again are different from the bathypelagic communities. So that was from the DNA. If we now add the RNA data into this and look at the active communities, so shown here in triangles, we see that the overarching pattern is pretty much the same. We see that the active communities are more similar both within these depth layers and also between the different depth layers compared to the DNA. And we also see that when we compare the DNA and the RNA from the same depth layer, that they are very different. And this was especially true from the mesopelagic and the bathypelagic layer, although they were significantly different at all levels. If we look at the biodiversity, here this is Shannon diversity. We see that the biodiversity was higher in the pelagic environment than in the sea ice. This was driven by richness. So there were more species in the pelagic. There was an increase by depth, so the highest diversity was found in the bathypelagic or mesopelagic and bathypelagic layers. But we also see that the basins were slightly different, and here the Amundsen Basin, shown in blue, had higher diversity than the Nansen Basin, which I think is opposite to what you found for the zooplankton. But this was only significant in the euphotic layer, and this was also true for the whole when you looked at the community composition. We go into the ecological functions, and here they are inferred from the taxonomic assignment. Then we see that we have, not surprisingly, phototrophs up here in the sea ice and in the sunlit ocean. And this beautiful, cute little thing here is a blue or a green algae, Carteria. It was quite dominant in the ice. Dinoflagellates were also most dominant here in the sunlit ocean, while parasites were more abundant in the deep and absent in the sea ice. And then the phagotroph, Cercozoa up here, and some environmental radiolarians here in the deep, they were in the ice and the bathypelagic. If we again add the RNA data, we see that it doesn't give exactly the same picture. So if you look at the pattern down here, you can see that this is actually quite different. So we see that parasites are much less abundant here in the active community. We see that we strangely have phototrophs even here in the deep community. And then we see these unknowns. They are also present here, but they're not as dominant as they are here. So here they make up to 10% or so of the sequence reads in this active bathypelagic communities. So we tried now for the final slide. I'll just show you a final figure. We try to look into this a bit more, and this is only the 50 most abundant species from the bathypelagic active community. And this is very preliminary, but anyway, it's a co-occurrence network. Oh, it's so tempting to move. And here each dot is a species, and it is colored according to which taxonomic division it belongs to. And and then these lines that are between them are co-occurrences. So if there's a very strong line here, it means that these species at the end of the line, they tend to co-occur in the different samples. So I want you to just focus on these yellow dots here, because they are the ones that were unknown, that we couldn't assign the ecological function to. And this is not surprising, because we can't even assign the taxonomy. We know that they are eukaryotes, but we are not able to go beyond that, so we don't know what they are actually. And what is interesting, I think, here is that if you look at these, you see that they form many connections, and they also have really strong connections, which suggests that they are central members of this active community here. So, final conclusion. So we saw that the communities were highly structured, and especially according to the layers. So they were different. I mean, the euphotic community differed between the two different basins, but this layering was much more pronounced, which is maybe not surprising, because we know that the Arctic Ocean is a highly layered ocean. The highest diversity was found in the deep layers and also in the Amundsen Basin. And then finally, I think maybe this is what I found most interesting, is that these active protein communities were so different from the total communities in the meso and bathypelagic. And this, of course, if we only look at DNA, which we often do when we look at communities like this, because they're too small to figure out in some other way, then we may be missing parts of this, the ones that are actually really important functionally. And then I also found it really interesting that we found these really abundant species that seem to have a central role and that we don't know what they are. So just to conclude, there's a lot of work to be done. Thank you.
Thank you, Anna, for a very interesting talk. And is one short question. Okay, Ove. I expected you will answer.
Thanks for the nice presentation. And I wonder if you include rare events in your diversity, especially if you work with RNA and DNA. RNA is more prone to lose rare diversity in the samples. Is that
we removed, I can't remember exactly the cutoff that we had, but we removed everything below a certain percentage during the process, the bioinformatic processing.
Have you cross-checked that?
And
many
then
of
we
those
also normalized so that it should be. Whether we have more species or
the
taxa
missing ones in the rare sphere. So that's our experience. In the rare sphere, we find the different ones between DNA and RNA. That's
okay, yeah. No, we didn't. I mean, for the... I think the rare ones, we've removed most of the rare ones.
Yeah.
But I mean, that could be interesting to look into to see whether there is a difference between. But I mean, they are very different, even independent of the rare ones. Okay. But it could be interesting to look into that as well. Yeah.
Thanks.
Thank you, Anna. Then I think we have to move on to Maxime. Let's see. Yeah, many things to be studying now, Maxime. So now I have to put my glasses on. I don't see anything. So Maxime Jeffrey, you are on the Marine Institute of Memorial University of Newfoundland. And you're going to spend 8 months, I heard. And you will now present this voyage or campaign for us. And we are looking very much forward to that. So please, the floor is yours.
So you might have heard about the Terra Polar Station, but there are different projects associated with that. One of them is a Norwegian project called Seasonality and Pelagic Diversity and Carbon Export within the Central Arctic Ocean, or SEDNA. Of course, we have many colleagues, international colleagues, associated with that project, and it's co-led by Marit Rekstad, who's based here. I will not teach you anything here, but Nansen drifted across the Arctic Ocean in 1893 to 1896 on board the Fram. He was ice-locked at the border between the Laptev Sea and the East Siberian Sea and drifted for a few years until being exposed in the Fram Strait, which of course created or launched the early stages of Arctic oceanography. Since then, even though it's not something that is discussed too much, there has been a lot of transatlantic drifts. This is a map of the transatlantic drift since the beginning with Nansen until the end of the 20th century. So there are still 26 years missing there. But see all these different paths. Most of them were conducted by Russian ice camps. Some of them were from American ice camps too. And since the past few decades, past decades or so, it's not possible to have an ice camp because of sea ice conditions. So there's now a new platform. It's called the Severny Polyus, or North Pole is a Russian vessel. But most of these drifts focus on geophysical research, atmospheric research, not so much on biology. And of course, there's an actual geopolitical context. More recently, or more within what we have as part of a team here, there was the 1997 to 1999 drift of Sheba, which was an American-Canadian partnership in the Canada basins on the Pacific side. They were on the Canadian icebreaker, the Groseilliers, and drifted in the Central Arctic Ocean. There was in 2006 and 2008 the drift from the Tara schooner, which reproduced more or less the drift from Nansen from the Laptev Sea / East Siberian Sea, the border, up to the Fram Strait. There were, of course, Mosaic in 2019, 2020, who did a drift from this location out to the Fram Strait. And some people here were part of the N-ICE project in 2015, which was north of Svalbard. So these were all drifts. So it's not, there are drifts in the Central Arctic Ocean, but all of them emphasize the fact that there's a need for longer-term, comprehensive, and seasonal observation. And I would add to that that in comprehensive, with a focus to some extent on biology as well. What's the link between the physics, the atmosphere, the different components, and the biology? And that's where the idea of the TerraPolar Station comes from. So it's a 26-meter round platform. It's a scientific vessel. It can propel itself. It can navigate by itself, 16-meter wide. It's very thick hull, 20 millimeters aluminum. So when we visited the shipyard, they had to find new engineering ways to actually be able to weld that thick aluminum on a vessel. It's really designed for science. So it's all built around a moon pool. And it's there's wet laboratories, there's acquisition rooms. So it's very a scientific vessel, but with a reduced crew of 12 to 18 crew members. So during the long overwintering leg, it will be 12 crew members. And then for the shorter legs in the summer and spring and fall, it can be up to 18 crew members. And then I will answer already one of your questions. So there's a moon pool and also possibility to dive and even have fancy Terra dry suit. The expeditions of the TerraPolar Station are, the concept behind it is to have 10 consecutive drifts that would cover the next 20 years or so. So the platform is not a research vessel, it really becomes an observatory. It's a drifting observatory that is there in the long term. The aim is to have that observatory for 20 years in the Central Arctic Ocean. Each of these drifts will be called the Terra Polaris. The first one is Terra Polaris 1 up to Terra Polaris 10, we hope. The first one will start in August 2026. It will stop in Tromsø here, so you'll have the chance to see it. It was here last summer when it went for its first sea trials in the Fram Strait. And the whole concept is really to have a holistic view, a comprehensive view of what's happening in the Central Arctic Ocean. So we will study the atmosphere. We have groups working on the atmosphere. There's a group working on sea ice, and there's a group working on the pelagic. There's also a long-term observatory group. There's a group that makes sure, of course, all these groups work together, but that makes sure that there's core observations where the methodology, the protocols are solid enough and then can be reproduced one drift after the other. So you would have a core set of, a program with a core set of variables that are measures, to which you would attach programs, different questions as we progress, because we don't know what the research question will be in 20 years from now. So it needs to have some flexibility as well. For the first one, there's a strong focus on biology, but as I mentioned, on the physical and how the physical environment impacts biology. And that includes the omics expertise from the Terra Ocean Consortium that have been developing over the past decades on the Terra schooner. There's a quite well-furnished lab on board, so that's on board the TerraPolar Station. There has been sea trials also on top of those in the Fram Strait. We were there in Finland, in the Gulf of Bothnia in December until January. There has been some tests for the meteorological balloons. Everything was very successful. There are a few things to improve, but the platform will be ready to go in August. As I mentioned, the idea is to have different components. So we have the atmosphere, we have the sea ice, we have the pelagic. So we need to scientifically found these different components. So we have one project, it's called SEDNA, funded by the Norwegian Research Council, that will focus on the pelagic part of the first program, the first drift, the Terra Polaris 1. We will study different aspects. So we'll cover physics. So we'll study the currents, the Atlantification of the Central Arctic Ocean, change in the water stratification. We'll see how this impacts the biodiversity. Again, with omics, we'll look into gelatinous zooplankton. We'll have zooplankton. We will have acoustics for fish distribution in among other, in the mesopelagic, under the ice. They will also look at into biogeochemical cycles. So we'll study how the stratification is linked to the nutrient fixation. We will study the carbon cycles also. So it's really trying to have an overview, a comprehensive overview of how the physical environment, how these different aspects of the water, changing water stratification, currents changes, impact the biology. And again, this is not an isolated project. That will be linked to other projects looking at the atmosphere and sea ice. We will have different activities. There will be different phases in this first drift. So we need to get into the sea ice. Of course, at the beginning, we'll not be able to have a proper ice camp. So as this platform gets stuck in sea ice, We expect some time where we can only work from the moon pool and deploy instruments from inside. But then as we consolidate the platform in the sea ice and we find a proper ice flow, the idea is really to start deploying instruments on the sea ice. So we'll have CTD chains, we will have ADCPs, we will have AZFPs, so acoustic zooplankton and fish profilers, and then we'll have sediment traps as well, long-term, short-term. We'll also have of course, the CTD rosette. There's a complete full CTD rosette, the same you will have on any other research vessels, where we can take samples for the omics, for instance. There's, as I mentioned, acoustics on board. There will be light measurements with light chains. We'll deploy zooplankton nets, different types. One trawl under the ice for macrozooplankton and potential ectoplankton. So you see, it's a whole suite of activity, the same you will have on a larger research vessel, but with a smaller crew. So the resolution, the time resolution might be a little bit lower to make sure that we have time to have all the operations. We will, as of now, we're working around a schedule that will be about 2 weeks for what we call one station, where we have a set of measurements. So why do we do that? What's the potential outcome? Well, there's a part on fundamental research. Some of the questions really relate to the origins of life. There's programs to look into the antifreeze proteins in sympagic organisms, which could be at the origin of ice linked to astrobiology, carbon cycles. There's also applied research linked to that. So we will, of course, put our findings and studies in the context of the Central Arctic Agreement on fisheries. There's a big component on biodiversity, and all of these, I think, segue pretty well with the previous presentation that showed that this is the kind of information is needed for the management of the Central Arctic Ocean. And there's also a part with students working on ecosystem modeling, and that will be tied to the climate modeling with the atmosphere team. There's also a strong outreach component. So the Terra Foundation has owned a pretty strong outreach component through the past decades with their work from the Terra schooner. And they would put to work the same people to the outreach on board the Terra Polar Station. And that's already ongoing. There's already activities with high schools, with different media. So pretty good coverage, which brings general attention to the Central Arctic Ocean. So that will benefit, I think, the whole community. And more of that, this project is linked also to Arctic Ocean 2050. So it's really, the idea is it's one additional platform to be linked to the other observation that will be there. And the idea is really to work as a complementary sampling platform and to develop joint projects. We already, for the SENA project in itself, so of course other groups have have more students and postdoc, but here with the project that is funded in Norway, we have 3 PhD students, Marion, Lucie, and Elise. They are distributed in different areas. They're not all based in Norway, but they are working together on that project. And we're also recruiting a postdoc with Marit working on biogeochemical cycles, if you know any good candidates. So there's many different institutions involved in that project, of course, and there will be more, I hope, in the coming 20 years. And I will let you on that picture from the first sea trials in the Fram Strait.
Thank you so much, Maxime. And you are also adjunct at UIT?
Yes, yes.
Yes. Forgot to say that.
That's how we got the project from.
Yes, reminded me. Yeah. Is there a quick question, very quick, because we have one more talk. It's a very exciting expedition.
I'll be around.
Yeah, he will be around. And then I think we will move over to the last presenter of today. And he will talk about those that we cannot investigate always in the summer, because they are quite active in the winter too. So, Davy Ford, you are from the University of Exeter, United Kingdom, and you will talk about colonus and trophic connections, and talk a little bit more about your study in the Fram Strait. So we're looking forward. The floor is yours.
Thank you very much for the introduction. I'm Davy Ford, a postdoc at the University of Exeter. I work within the Ocean Plankton Research Group led by Daniel Mayer, and the main purpose of this presentation today is to showcase the potential role of Calanus copiopods in between surface waters and the deep sea in the Arctic Ocean, which will hopefully underscore one of the many reasons why it's so important to understand how these populations may be changing in the future. I wanted to just start the talk by thanking all of the amazing co-authors that were involved with this research long before I was involved. So for context, I'm an experimental physiologist. I mainly work in the lab within the confines of the UK. And it's really the names you see on the screen at the moment that did most of the hard work for this research, including the data collection and the conceptualization. So onto the project itself, as I'm sure many of you are well aware, Calanus copiopods dominate the mesozooplankton biomass in many Arctic regions, where they play a pivotal role as important prey for many seabirds, mammals, fish, invertebrates, etc. They also play key roles in the marine biological carbon pump, where we know they export substantial quantities of carbon from the surface waters to the deep sea, where it can remain sequestered for hundreds of years, depending on the specific mechanism of carbon transfer and release. The functional importance of Calanus copiopods is largely dictated by their life cycle dynamics. So in the Arctic, they typically start their life cycle in spring. They'll gorge themselves on the vast availability of photosynthetic material, and they'll use the assimilated material to synthesize these vast lipid pools that can take up most of their biomass. They specifically store lipids in the forms of wax esters, which are comprised of one fatty acid chain and one fatty alcohol chain joined together by an ester bond. They're also known to specifically biosynthesize large quantities of 20:1 and 22:1 fats. And this is a unique biosynthesis pathway, and it's very useful in a trophic context, because it means if we look into the lipids of other Arctic animals and we find high abundances of these fats, it's a very strong indication that those animals have been feeding on Calanus. At the end of summer, Calanus will then migrate down to the deep ocean, usually somewhere between 500 and 2,000 meters deep, where they overwinter in a dormant state known as diapause. And in undergoing this migration, they inject vast quantities of carbon from the surface to the deep sea. Whilst Calanus are in diapause, they will fuel their metabolism with their lipid stores. So they will release carbon in the form of CO2 via respiration, but they will ultimately return to the surface the following spring with roughly half of that carbon in their lipids still intact. Alternatively, if they die during diapause, the carbon in their lipids is assumed to be remineralized by bacteria. However, if Calanus are predated on by deep-sea animals, then the surface-derived carbon in their lipids could be directly transferred into deep-sea benthic food webs. And this has implications for carbon sequestration estimations, but it also implies that Calanus could completely modify the typically very food-limited trophic landscape of deep-sea ecosystems by bringing down with them these vast quantities of really nutritious and fatty materials. Despite this potential functional role, we know very little about the trophic connections between diapause in Calanus and deep-sea animals, or whether these connections even exist. So we had quite a simple aim, and it was to investigate the potential trophic connections between Calanus and deep-sea benthic animals. So we were specifically focusing on animals found on the sea floor. With that in mind, the team set out to the long-term ecological research observatory House Garden in the Fram Strait between Greenland and Svalbard, and this research was conducted in 2023, so it's quite recent still. And the nuts and bolts of the methods involved conducting parallel plankton net deployments to collect Calanus from between 1,000 meters to the surface, with epibenthic sledge deployments, which you can see in the photo here, in order to collect deep-sea benthic invertebrates, typically below 2,000 meters deep. And here we have some examples of the taxa we analysed, which includes amphipods, polychaetes, mysids, and the two most abundant species of mesozooplankton found in the Fram Straits, Calanus vimarchicus and Calanus hyperboreus. The team then measured a suite of different metrics, but for this talk I'm just focusing on, did we find those Calanus fatty biomarkers in the lipids of the deep-sea benthic fauna? Having said that, the first key result to know is that we did actually collect Calanus copiopods with our epibenthic sledge deployments at 1,500 and 2,500 meters deep, despite the fact this study was conducted during the summer. So this indicates two things. It indicates firstly that not all diapause in Calanus may return to the surface. They may just fail to return. It also indicates that Calanus could be on the menu for deep-sea benthic fauna because they were indeed found near the seafloor in the deep sea. And then here we have some multivariate analyses, and I'm not going to go into too much detail here. I'm just going to highlight some key differences between Calanus and benthic taxa. So, for example, if we look at the lipid groups, we can see that Calanus in red were associated with saturated and unsaturated fatty alcohols. This makes sense. As I mentioned earlier, their wax esters are comprised 50% of fatty alcohols, whereas most other animals just store fats as fatty acids because they don't need the functional roles that alcohols provide in buoyancy. But then when we look at the fatty acids and the fatty alcohols, we can see quite clearly that Calanus were associated with 20:1 and 22:1 fats. So no surprise, Calanus were associated with Calanus biomarkers. But when we look at the relative abundances of these biomarkers, and the bottom plot here shows the total abundances of the biomarkers, we can see they are present at moderate to high concentrations within all of the benthic taxa analysed. So if you look at the mysid shrimp, for example, you can see that they were roughly a quarter of their lipid pool were Calanus biomarkers. But even in polychaetes, which were mostly found below 5,000 metres, about 20% of their lipid pool were Calanus biomarkers. And we have some large variability in the amphipods, for example, and this is largely due to species-specific trends, which I can answer some questions on if you'd like. And I won't talk about the isotope data much, other than just highlighting that the taxa with high abundances of Calanus biomarkers were roughly 1 trophic level above Calanus. And again, you have these species-specific trends, particularly in the amphipods, which are known to be very diverse in their feeding strategies. So the key results then, or our results suggest that deep-sea benthic animals are feeding on diaportin Calanus and assimilating their biomarkers into their own lipid pools. This then in turn suggests that a portion of the carbon brought down to the deep sea via the seasonal lipid pump is actually transferred directly into deep-sea benthic food webs, which in turn indicates that Calanus could play really important roles in supporting the integrity of these usually very food-limited ecosystems. There is a slightly more sinister implication for our results regarding the potential transfer of anthropogenic contaminants. So many contaminants are known to be highly lipid soluble and are routinely found in lipid-rich zooplankton in the Arctic. So if benthic fauna are feeding on diaportin Calanus, then those contaminants could essentially be shortcut down from the surface into the deep sea. And this may become increasingly relevant following the growing industry activities within the Arctic region. So to summarise then, we think our results provide evidence to suggest that Calanus play important roles in the trophic connection between surface waters and the deep sea, which kind of underscores one of the many reasons why better understanding the functioning of their seasonal lipid pump and how this functioning could change in the future is really important to predict the future integrity of Arctic ecosystems. So thank you everyone for listening, and thank you again to the amazing co-authors. This paper is currently in second review in Deep Sea Research Part 2, so please do keep an eye out for it, or pop me an email if you're interested in if and when it's published. And that's all from me, so thank you very much.
Thank you so much. Very, very interesting. And I think we have overseen this lipid pump, even if it's been introduced for a few years back. And we have seen in our cruises in the Arctic Ocean high abundances of Calanus hyperboreus in the surface 50 meter in summer. So it's a massive biomass, I would say. Yeah. Any questions? Yes, Håkon.
Thanks. Super interesting. What about fecal pellets?
Yes, good question.
What happened to them?
So the potential, we discussed this in the discussion, there are various potential routes by which these biomarkers are found in the benthic taxa. Fecal pellets, molts, other animals that feed on Calanus that die and sink, or live Calanus. And we break down each of these different reasons where we think the evidence points towards. The main thing with fecal pellets is that low in the Fram Strait, and fecal pellet abundance at the depths we were studying is quite low. And B, Calanus fecal pellets contain much lower concentrations of these biomarkers relative to their lipids. So in order for these animals to be eating fecal pellets and getting those biomarkers, they'd have to be eating an enormous amount of these fecal pellets in order to get those concentrations. The one caveat is that we don't know the fatty acid turnover rates. So there is a chance that they are selectively retaining these fatty acids, and that's maybe how they build them up. But the available evidence points towards the idea that they're feeding directly on calamus, which is not to say the other things aren't happening, but I think future research looking at, for example, gut content analyses and stuff like that would really shine more light on this.
Thank you so much. I think we will close the session here. And question there, Håkon. We also notice a very high, first an applause. It's a very high mortality winter-spring transition on these colonies. So they are non-consumptive mortality. So they probably, where do they go?
Yeah, well, that's what we think that we don't know how many of them eventually then come back to the surface. What happens if they fail to do so?
Yeah, no resources left and so on. Super interesting. So just to conclude, we are in the progress of knowing more, and I see there is a lot of enthusiasm and expeditions the coming years. We have the Arctic Ocean 2050, we have the Terra Polar, and there are several other coming in. And I think there also will be this open call in 2027 for at least the Arctic Ocean 2050 and UK potentially. And Germany are coming after to provide same or some kind of similar calls. And then, of course, we also have IPI 2032, 2033. So we are in a very exciting decade. So looking forward to see you again on next conferences and learn more. Okay, thanks for all the presenters. And tomorrow it will be indigenous knowledge in interesting panel debate, but with some talks before in the afternoon. So I think it's 13:30. And then we also discuss more what is our knowledge? Can it be used? Be useful? We have learned about that for the previous session. Okay, thank you.